Hello everybody. I'm very happy to be here today and present you my work on cell type specific vulnerability to traumatic brain injury. I'm Araks Martirosyan from the Laboratory of Glia Biology at Center of Brain and Disease Research located in VIB-KU Leuven in Belgium. And this work we are doing in collaboration with the University of Amsterdam and the Bioinformatics CRO. So before going on with the main topic of my talk, I would like to give you a brief introduction on cell identities and the principal dogma of molecular biology. As we know, in our organism, all the cells are differentiated from stem cells, but they get different identity during their maturation, and they are located in different organs and perform totally different functions. How different muscle cells and nervous cells could be, you can imagine. So what is making these cells different? Going to the principal dogma of molecular biology, we have all the different cell types with different morphologies, and they are sharing the same DNA. What is different, what is making different these cells, is their mRNA content, which is transcribed from their DNA. So only a specific set of mRNA is transcribed in every cell type, and they are synthesizing or being translated to different types of proteins. And these two steps are making cells very different. These are the proteins that are performing different functions in the cells. They allow them to interact with each other. They are defining their shape, and so on. So the main difference is coming from the mRNA content in various cell types. So what is revolutionary about recent technologies, single-cell transcriptomics technologies, is that nowadays we are able to quantify the whole transcriptome in a single cell. This is giving a lot of advantages, and the principal advantage is that we can quantify transcriptomic changes in case of injury and disease, something that has not been possible before. And nowadays we are doing this at a single cell resolution, which is of course giving us an information about the functional molecular pathways in a very cell type specific manner. So we know in which cell types which pathways have been altered in the case of injury or disease, and therefore we can design very effective therapeutics that is targeting given cell types. So these revolutionary technologies came in use approximately 10 years ago, and now they are revolutionizing the biomedical research. In this particular talk, I would like to tell you how did we use these technologies to understand molecular changes happening in the brain after traumatic brain injury. The traumatic brain injury is caused by an external force which is affecting our brain function. And it is a major global health concern. It is affecting approximately 69 million people per year, and we are all vulnerable to that. Traumatic brain injury can occur in situations like falling, traffic accidents, and so on, impacting people all around the world. So, of course, the first cell types that we want to look at in this case are neurons, which are the building blocks of our brain. And in the brain, there are different types of neurons, and they interact with each other by transferring signals from one to other and creating a network of communication. Thanks to this neural network, we are able to think, reason, memorize events, and so on. However, neurons are not the only cell types in the brain. There are also glia cells that my laboratory is focusing on, and in particular, we are studying astrocytes. So in this presentation, I'm going to focus on astrocytes alone. However, there are other glia cell types in the brain. So astrocytes are star-shaped cells, as you can see on this slide, and they are known to support neuronal functions, neurogenesis. They are helping neurons to develop, and they support neuron-to-neuron communications at the synapse. So what is happening to these cell types after injury? Neurons can be damaged or lost, so they are not able to perform their normal functions anymore. And this is, of course, unwiring the neural network. So neurons are not able to communicate anymore. However, again, neurons are not the only cell type that are affected by traumatic brain injury. Astrocytes also respond to that, and they become reactive. By becoming reactive, astrocytes change their identity, they change their shape, and they lose the capacity to perform some functions, and they gain new functionality. So this concept is very well described in a recent publication by Escaltin et al. In Nature Neuroscience, where authors try to summarize and define all about reactive astrocytes. So if you're interested to know more about this process, I would recommend you to read the paper. So in the result of this process is happening in the brain, of course, the brain functionality is damaged. What is interesting about traumatic brain injury is that it is resulting in long-term cognitive impairment in 50% of cases. And this is telling us that something is happening in hippocampus, which is the brain region responsible for cognitive functions, memory, and thinking. Let me switch to the mouse model, the model that we study to understand molecular changes in the brain after traumatic brain injury. So here you can see a mouse and a slice of a mouse brain taken from Allen Brain Atlas. So the mouse brain, here you see stainings of neurons there, and you can see that it is well separated into different regions, and these regions are well defined. At the top, we have the cortex. And usually in case of traumatic brain injuries, it is the cortex which is impacted first. However, the cognitive impairment is also related to the functions of hippocampus. This is the inner structure here. You can see hippocampus is well recognizable in the brain. It has a very particular neuronal structure. So, what is happening in the case of traumatic brain injury? Let me show you the model that we use, which is the controlled cortical impact model. And here you can see the mice who is undergoing a TBI surgery, and during this surgery, the mouse head from the cortical side is hit. And you can compare here on this slide brain of a control mice with the brain of the TBI mice. And you can see that the cortex, the upper part of hippocampus, is highly impacted by TBI. The tissue is almost lost. Hippocampus itself doesn't suffer the loss of tissue. However, when we put these mice in a condition to test their memory and how well they can memorize spatial information, we see an impairment of their dysfunction. Here I would like to show you the spatial memory test that we performed, and I thank my colleague Pascal Belfield for performing this experiment and quantifying the data. So what we do, we put a mouse in a water pool, and in that pool there is a hidden platform. And mice repeatedly perform this function. They are in the pool, they swim until they will find the hidden platform, and they will rescue. So day by day, mice are supposed to learn the shortest path to the hidden platform. When we do this experiment with control and TBI animals, 5 days of experiment is showing that both the mice improve in their performance. However, there is a significant difference between the performance of TBI and control mice. And TBI animals usually memorize the location of the hidden platform much worse than the control animal. They need more time to find it. This is showing, so spatial memory is directly linked to the function of hippocampus, and this is indeed showing that in this model, although the hippocampus itself didn't suffer a loss of tissue, in long term it is leading to the loss of spatial memory. Let us go a little more into details and understand the structure of hippocampus and what is going wrong, what is the reason of this deficit of memory. Zooming on hippocampus, we can see a nice neuronal layering, and here I highlight the region of dendrite gyrus. What is interesting about the region of dendrite gyrus that it is a source of stem cells in the brain. It is one of the positions where in adult brain we can still find stem cells. And they are located in this particular zone, which I show by the arrow, sub-granular zone. So when we try schematically to present what is the structure of this sub-granular zone, here on the left you can see by colors cells that are young progenitor or neural stem cells, and they are located, residing in this sub-granular zone. These cells are able to differentiate to neurons, and this part of differentiation is well investigated. In adult mouse, it is reported that in the adulthood, neurogenesis continues in hippocampus, and this is maintaining the normal function of the brain. Here you can see the developmental part of neurons. So neural stem cells, which are in yellow color in this particular illustration, slowly they start developing processes, and they generate mature neurons which integrate to the molecular layer of hippocampus and replace damaged cells. Most curiously, it is known that astrocytes are sharing these progenitors with neurons, so they are differentiated from the same neural stem cells and progenitor cells. The developmental part of astrocytes is less understood, it is less studied, and it is one of our goals to understand how these cells mature and how effective is astrogenesis in adult brain. So when looking into the literature and reading the impact of TBI on hippocampus, we see reports that are showing that in hippocampus the neurogenesis is activated itself. However, these newborn neurons have dramatic morphological abnormalities. Their shape is different, and therefore they are probably maladaptive to the neural circuit. And these are, of course, processes that we need to understand to configure it better, so that newborn neurons would integrate into the neural circuit and assure normal function of hippocampus. So to confirm these reports, in our hands, we did some extra experiments. Here you can see the stainings that we did in TBI and control mice by the DCX marker, which is a marker of young neurons and progenitor cells. On the right, you can see the stainings and the quantification of it. So what we observe is that in total, the number of DCX cells is increasing in TBI condition. And this increase is mostly dominated by the cells that are in their developmental stages C and D. You can see in this illustration that the developmental stages C and D are the points of differentiation when small short processes are already present. However, these are not mature neurons yet. So the question is, why is the neurogenesis blocked in this stage? It is locked in this stage when we have only short processes and it doesn't develop further. Next, we looked also at astrocytes after mice undergo TBI surgery, and GFAP is a marker of reactive astrocytes. So when staining for this marker, we saw that on the side, on the hemisphere of the mouse brain, where we had the injury, the GFAP expression is much higher, which is indicating about ongoing astrogliosis, which is the process of generation of reactive astrocytes. So indeed, we did know that young neuronal progenitors and astrocytes are affected in hippocampus after cortical traumatic brain injury, and we want to understand the consequences of that. So our first research question is to understand how TBI is affecting neurostem and progenitor cells at the molecular layer. And our second research question is to understand how does cell type positioning alter in hippocampus and the surrounding tissue after TBI. To answer to the first question, we used single-cell whole transcriptome sequencing as I was describing in the beginning of my talk. So what we did, we took a sham control and a DBI animal. We dissected dendrite gyrus, the region where we expect neurostem and progenitor cells to be located. We extracted with specific sorting of these NSPC cells and put them into a microtube. This is a workflow suggested by 10X Genomics, and it is working very well, becoming the gold standard. Pulling these cells out of the dendrite gyrus and into the microtube, we are able to separate them one by one, extract their mRNA content, do sequencing, and get information about the whole transcriptome of these cells. In my next slide, I will show you our results. So here in this slide, I'm showing all the cell types that we were able to identify in the brain based on their marker gene expressions. And if you are not familiar with this plot, let me quickly introduce that to you. So here we have a UMAP plot where each dot is a cell, and cells similar to each other are making a cloud. So each cloud is a cell population. As you can see here, we have found a number of cells in dendrite gyrus. So cells, glia cells like microglia, oligodendrocytes. We have found neural cells, endothelial cells, oligodendrocyte progenitor cells. But here in this presentation, I want to focus on the two lineages, one representing the development of astrocytes and the other presenting development of neurons. Here on this plot, you can see the astrocytic lineage in green color named A1, A2, A3, and A4, and the neuronal lineage with blue color N1, N2, N3, and N4. You can notice that both these lineages are initiating from neural stem cell and radial glia-like populations, as it is expected, because we know that astrocytes and neurons are sharing progenitors. So this is already very nice to see in adult brain the whole developmental path of astrocytes and neurons and their initiation point from the same population of neural stem cell-like cells. So here in this plot, we have all the data put together, both DBI and control animals. And when we look into the origin of these cells, you can see the bar plot here, where the red color is representing the percentage of cells coming from control animal, and blue one is representing percentage of cells coming from DBI animal. You can notice that astrocytic lineage named A1, A2, A3, and A4 is suffering loss after DBI. So after DBI, premature astrocytes die or they change their identity. And what comes to neurons, at least one of the populations, we saw a significant increase in numbers after DBI, and this is the population N4. Consistent with our previous observations, we indeed have an activated neurogenesis, and the second interesting observation that we make is that astrogenesis is either suppressed or astrocytes die after DBI. So now let me switch to the second research question that we were trying to tackle in this project, and it is about cell type positioning in the tissue, in the whole hippocampus and surrounding tissue, and how does this alter in TBI conditions. We couldn't extract this information, of course, from the single cell transcriptomics database that we had, because there we are dissociating the tissue and we are pushing cells out of the tissue. So we don't know from which exact spatial locations they are coming. That's why we need to complement our database with spatial transcriptomics. So when it comes to spatial transcriptomics technologies, it is about quantifying transcriptomes of the cells in the tissue without touching the tissue itself. In here, we have two possibilities currently. First one is to obtain the whole transcriptome from a given site of the tissue. However, the resolution of the whole transcriptome in situ sequencing technologies is low, and we cannot obtain information about single cells. So we know that this transcriptome is collected from a given location in the tissue, but we don't know if in that location there was one cell, two cells probably, or maybe it was a gap between cells. So we are not very sure that every dot is representing one cell. And the second set of technologies that are available are allowing to quantify at a single cell resolution transcripts. However, the number of transcripts is limited. Until recently, it was limited to 10-12 markers. However, recent technologies are allowing to increase this number significantly. And our choice was to go for this because we were interested in single cell quantification of the transcripts in the tissue. And thanks to VIB TechWatch funding, we had access to the technology of Resolve Biosciences before it was commercialized. So this technology is allowing to stain 100 markers at the single cell resolution at the same time in the tissue. So let me show you brains that we stained. So we did our first stainings were on 2 control and 2 DBI brain sections. And since we had the capacity of 100 genes, we chose differentially expressed genes between NSPC cells to identify, to find back these populations in the tissue. So we chose NSPC markers. Next, we chose mature astrocytic and neuronal markers in order to see what are the changes at the level of the tissue. I will quickly show you our results. We were able to quantify these transcripts, our transcripts, 100 transcripts at a single cell resolution. And getting back to the expected picture of young progenitors, neural stem and progenitor cells, we indeed found them in the subgranular zone of hippocampus. And here you can see our quantification. So dark blue dots are showing more NSC, neural stem cell-like populations. Yellow cells are mature astrocytes or neurons, depending on the lineage. And you can notice that indeed blue cells are found in subgranular zone. Astrocytes develop towards hilus, and neurons develop towards granular neuronal layer of dendrite. This is an expected picture, and it didn't change from control to DBI animals, which means partially this pattern was not impacted by DBI. Pattern itself was not. Going larger to the level of the whole tissue, we were able to recapitulate the neuronal structure of hippocampus. Here you can see the hippocampal layering, and on the side, our clustering analysis based on 100 marker genes, and you can see that we have found neuronal layers that are colored in various colors here, neuronal layers in dendrite and in the surrounding zones. Then we looked at the populations at the hippocampal level. So we don't have a single cell 10x dataset representing the same frame of the brain. However, based on our 100 marker genes, we saw very interesting shifts after DBI. Here you can see a comparison between control and DBI animals, and in black dots you see a cellular population which is representing reactive astrocytes, and you can see that in control animals this population is missing, while in DBI animals we have a very specific spatial pattern where these cells are found. This is very interesting, and it is showing that not all astrocytes become reactive. They are responding to injury to spatially distinct way. Only a fraction of astrocytes in very specific positions become reactive. And the next observation that we have is the distribution of neurons, and for some populations, some neuronal populations, we saw the dropout of mature neuron numbers. In this particular case, you can see in the surrounding tissue of hippocampus, we have a dropout of neurons, and in TBI condition, we observe less cells. With this, let me go to my conclusions. So I hope I could introduce you the concept of single-cell and spatial transcriptomics and how this could be used to study injury in disease. I showed you that neurogenesis in dendrite gyrus is impacted after TBI, that distinct associated progenitors have been found in our analysis in dendrite gyrus, and the number of these young astrocytes is dropping after traumatic brain injury. I showed you a population of reactive astrocytes which was occurring spatially very locally in the sites where the injury has happened. And finally, I showed you that we lose some neural populations in the parts of the brain where we had astrocytic response. So in the future, we are going to make use of our whole transcriptome single-cell database to identify molecular pathways that have been affected by TBI and to understand what is going wrong with neurogenesis and astrogenesis in the brain after the injury. And secondly, we are going to enrich our spatial database in order to understand the relationship between neuronal loss and the appearance of reactive astrocytes. With this, I want to thank you all. I want to thank my colleagues who contributed in this research and our funding agencies for making it possible. Thank you very much for your attention.